Okay, welcome guys again to another lecture of biophysics. We are going to discuss about biomedical application of electrical or electric potential difference in the cells. Conduction of electrical signals in neurons. Okay, neuron. We are going to discuss about conduction of electrical signal through neuron. Okay. So, if you are new to this channel, make sure you subscribe to my YouTube channel and hit the like button, since you are going to get valuable knowledge at an affordable way, which is very cost effective as well as time saving. Uh, so, if possible, you can also share to people and make sure you stay tuned with coming lecture. For that, please consider subscribe. I'm going to share lots of information about biophysics. It is the physics with integration of biology concept. What it is the result of it? Exception. Okay, just kidding. Okay, let's start from the beginning of it. Since we are going to discuss one by one, line by line explanation of it. So, the human nervous system is remarkably for its ability to transmit information in the form of electrical signal. This signal are carried by the nerve, and the concept of electric potential difference plays an important role in the process. For example, sensory information for eyes, ears is carried by the brain to the brain by the optic nerves, auditory nerves, respectively. So we all know that for our processing of any messages, it collects the nerve through auditory images and goes to the brain. Okay, for that it is auditory nerve, and for optic it is optic nerve. So other nerves transmit signals from the brain or spinal column to muscle. There are also some nerves that transmit signal from brain to spinal column to muscles, causing them to contract. Okay, so when we contract, there is some exchange of, we already know this, there is a exchange of polarization at the muscle level. It can be calcium, it could be potassium, it could be sodium. So in that case, these are the dendrites here, you can see, and this is the cell body, and this is the nerve, and this is the axon. And we all know that every information transmitted from certain, suppose essence of, when you touch a hot appliance, it signals through the neuron and transmit to other neuron. So it works like that. So it indicates that other nerve transmit signal from brain to spinal column or muscle, causing them to contract. Still, other nerve carry signal within the brain. Okay, after it is up to nanosecond, which is quick, very quick for the processing. It goes to brain first, and then after quickly interpreted by the brain, it signals to remove the hot appliance, like that. So it all happened in a microsecond. So what can I say is that it functions, or our body functions in a very complex way. So it is our honor to aware of this mechanism. All right, so let's continue. A nerve consists of bundle of axon. Axon is there. It can be myelinated in some cases. Myelinated means in this shape, but it can also be unmyelinated. It depends on types of axon, types of neuron, and each axon is one part of nerve cell or neuron. A figure illustrates that neuron consists of cell body, numerous extensions called dendrite, and a single axon. And single axon is here, and this is the cell body, and this are the dendrites, and this is nerve ending. The dendrites converts stimuli. This collects the stimuli such as pressure or heat or electrical signal travel through this neuron. The axon sends the signal to the nerve ending, which transmit to the signal across the graph called synapses. There is a synapses here between one neuron to another neuron. There is a place called synapses. Right. So this is synapses here. The fluid inside a cell, intracellular fluid, is quite different from the outside the cell, extracellular. Both fluid contains concentration of positive negative ion. Suppose let's say this is a cell as compared to outside. Suppose this is a cell having potassium ion inside and sodium ion outside. This is extracellular and this is intracellular. Okay, so we know that for its exchange of sodium and potassium pump through potassium pump, it requires ATP. Then it transfer 3 sodium ion outside and it transfer 2 potassium ion inside. So the fluid inside cell, intracellular fluid, is quite different from the outside cell, extracellular fluid. Both fluid contain concentration of positive negative ion. However, the extracellular fluid is a rich sodium ion, as I've just told you, and chlorine also outside the extracellular. And where the intracellular fluid is rich in potassium and negative ion, protein or amino acid. This concentration difference between the fluids are extremely important for the life of the cell. Alright, if the cell were freely permeable, means we know the transmission of any ions or any form of substance through cell could be two way, which is passive, and another is active transmission of ion core. So if it is passive, there will be no ATP required. If it is active, then it requires ATP. And for passive, the transfusion must be along the concentration gradient, and for active, it must be against. So it requires ATP or some proteins so as to pass the ions. So if the cell membrane were freely permeable, the ion would diffuse across it until the concentration on both were equal. This does not happen, although. Because a living cell has selectively permeable membrane, ion can enter or leave the cell only through membrane channel. And the permeability of the channel varies. Markedly from one ion to another. For example, it is much easier for potassium ion to diffuse out of the cell than it is for sodium to enter the cell. As a result of selectively permeability, there is a small buildup of negative charge just on the inner side of the membrane and equal amount of positive charge on the outside. So we can clearly know that inside the cell we have much negative charge as compared to outside, but for extracellular we can consider that there are more positive charge outside the cell. Okay, the buildup of the charge across the buildup of the charge occur very close to membrane. So the membrane acts like a capacitor. So now we are going to implement the physics concept here. Since this is biophysics, we are going to discuss both physics and biology. So the buildup of charge occur very close to membrane. So the membrane acts like a capacitor. So we know that if it is inside the cell, it is much negative charge here, and outside there is much positive charge. Then we can presume that there is two capacitor like that. So like a capacitor, elsewhere In the intracellular and extracellular fluid, the equal number of positive negative ion, so the fluid are overly electrically neutral. If it's positive and the negative charge are what can I say, suppose this is extracellular, suppose this is intracellular. If the positive and intracellular are of similar magnitude, means in a sense that same value, suppose 20 volt 20 coulomb, let's say 20 coulomb charge of positive and 20 coulomb charge of negative, then it certainly going to neutralize, and it becomes electrical neutral overally. Such a separation of positive and negative charge gives a rise to electric potential difference across the membrane called the resting membrane potential. So, such a separation of positive negative charge gives rise to electric potential difference across the membrane called the resting membrane potential. The resting membrane potential ranges from 40 to minus 90 volt. In a typical value, minus 70 millivolt, the minus sign indicates that the inner side of the membrane is negative related to the outer membrane. Alright, so the concept is much simple, that in some certain cases there is a difference between the two position, extracellular, intracellular. So it says that everywhere in the intracellular, extracellular fluid, there are equal number of positive negative ions. So the fluid are overly electrical neutral in certain cases, and in certain cases, such as separation of positive and negative charge give rise to electrical potential difference across the membrane called the resting membrane potential. In neutron, the resting membrane potential ranges from minus 40 to minus 90 millivolt, which is typically minus 70 after. It must be positive. So okay, so the minus sign indicates that the inner side of the membrane is negative related to outer, so this should be positive. So let's discuss some picture representation. So let's suppose this is extracellular cell and this is intracellular, and there is a cell membrane here. One thing you should know, that cell membrane is made up of phospholipid. Phospholipid means that I will not come to the chemical structure in detail, but you should know that phospholipid structure, the outer membrane is water loving, which is hydrophilic, and in a sense of lipid, we can say lipophobic. But inside this, these are hydrophobic but lipophilic. Alright, you should know the terminology. Let me write it for you. Outside membrane is called to hydrophilic or lipophobic. And inner membrane is this one, I mean, is called hydrophobic or lipophilic. Okay, let's discuss again. Extracellular fluid is this portion and intracellular is this portion. Let's presume that. So we know that intracellularly we came to know that it has more negative charge inside as compared to extracellular, which is much positive charge. Alright, so if we made a channel gated membrane made up of protein or any sort of, we can easily pass through it. Let's see what the concept actually means according to physics. Membrane capacitance means positive and the negative charge layer forms on the outside and the inside surface of the membrane during its resting state. Okay, there is positive charge Q, there is a negative charge Q, so there is a basic difference between the two charges. So it creates a membrane capacitance in a sense, and this lip what can I say lipophilic or hydrophobic layer must be the dielectric substance or material. And after that, let's discuss membrane resistance. Is when a stimulus is applied to the cell, positive sodium ion rush into the cell, causing the interior surface of the membrane to become momentarily positive. So this the what can I say the basic idea is that membrane resistance, when a stimulus is applied to the cell, stimulus means a just like a what can I say response. When a response is applied to the cell of positive charge, which is sodium ion rush into the cell, causing the interior surface of the membrane to become momentarily positive. So whenever, let's take an example of contraction of muscle. I won't go to detail, but whenever we try to contract our muscles, it quickly, while we doing a bicep curl, it exchanges the charges quite fast, I must say. So like by taking that example, when a stimulus is applied to the cell, positive sodium ion rush into the cell, causing the interior surface of the membrane become momentarily positive, and just for nanoseconds, and after that back to the position. So there's a quick exchange of charges between, if positive is momentarily entered to intracellularly, then the negative must be exchanged to extracellularly momentarily also. That is the main idea of it. Let's discuss further the physics of action potential. So this is a very important graph quotation here. The physics of an action potential. The resting neuron, let's say, is one of that is not conducting a electric signal. The change in the resting membrane is the key factor in the initiation and conduction of a signal. When sufficiently strong stimulus is applied to a given point on the neuron gets in the membrane open and sodium ions flood into the cell. The sodium ion are driven into the cell by attraction to the negative ion on the inner side of the membrane, and well as the relative high concentration of sodium ion outside the cell. The last influx of sodium ion first neutralize the negative ion on the interior of the membrane and then causes it to become positively charged. As a result, the membrane potential in this localized region goes from -70 millivolt, the resting potential, to about -30 millivolt in a very short time. The sodium gets then close to, and the cell membrane quickly returns to its normal resting potential. This change in potential from -70 millivolt to -30 millivolt back to -70 millivolt is known as action potential. The action potential lasts for millisecond, and it is the electrical signal that propagates down the axon, typically at a speed of about 50 meters per second, to the next neuron or to a muscle cell. So I will explain this concept. Let's look the graph once again. So action potential lasts for some certain period of milliseconds, but membrane potential is the potential developed at the membranes. It is a charge separation of the extracellular and the intracellular portion. So it is going to be traced with the time and with its magnitude with this graph here. Sodium influx must be of the highest magnitude here, and resting potential is up to this few instance. So a resting neuron is the one that is not conducting an electrical signal. If we suppose this is the neuron membrane, and the resting neuron is one of that is not conducting any electrical signal, so that is the resting phase. And change in the resting membrane potential is the key factor in the initiation of and conduction of a signal. So when a sufficiently strong stimulus is applied to a given point on the neuron, neuron gets, let's say, in the membrane open and sodium ion flood into the cell. The sodium ion are driven into cell by attraction to the negative ion on the inner side of the membrane, as well as the relative high concentration sodium ion outside the cell. So this is what the actually means is that the sodium ion are driven into the cell by attraction to the negative ion on the inner side of the membrane, as well as the relative high concentration of the sodium ion outside the cell. The large influx of sodium ion first neutralize the negative ion on the inferior of the membrane and then causes it to become positive charge. As a result, the membrane potential in this localized region goes from minus 70 millivolt, the resting potential, to about minus 30 millivolt in a short time. The sodium gets then closed, and then the cell membrane quickly returns to its normal resting potential. The change in the potential from minus 70 to minus 30 millivolt and back to minus 70 is known as action potential. This must be action potential. This magnitude must be action potential. The action potential lasts for a few milliseconds, and then in electrical signal that propagate down action, typically a speed of about 50 milliseconds to the next neuron to muscle cell. You know that's action potential with the graph of action potential and the membranes below here with the time axis on x-axis and y-axis as potential below. So first thing first, make sure you subscribe to my YouTube channel and hit the like buttons, and if possible you can share my video, and it's all up to you, but stay tuned with my lecture series so then you won't miss any updates on further lecture. So into this episode we particularly going to discuss about the exchange of ion through membrane across the cell membrane with the physics notation of action potential. So we are going to discuss the diagram here. So first thing first, this segment is for depolarization, and this segment is for repolarization, and this is depolarization. Depolarization means the contraction, which is if it is for cardiac we can consider it as systole, and for repolarization for in a cardiac muscle it can be considered as a repolarization. So this one is the depolarization, which is in the phase of contraction with maximum amount of contraction attains to this almost this peak, and then after that after reaching to the maximum limit it again comes to the repolarization, which is diastole for cardiac relaxed state, and sometimes it goes to even resting potential like in our further as we know the respiration terminology of inspiratory reserve valve, like that some external some extra, let's say extra resting state. So in those cases we are going to use the term resting potential. So that's the only thing where I want to share of this on this graph. So the action potential is caused by the rush of the positive sodium ion into the cell and subsequently return to the cell to its resting potential. So sodium ion, I think you have all come across to my YouTube lecture series, so you probably know the exchange ion of in the intracellular portion and extracellular portion. So as we all know, there's sodium ion. Just I want to remind you so that you can also cope up with it For those of you are not newly joined, who newly watched in this channel, make sure you subscribe. And for now, you have to focus on this, but after that, you have to go to the previous lecture so that you can grab the idea of it, of biophysics. So for now, let's see. This is intracellular. Let's suppose this is a cell. As compared to positive, the magnitude of positive charge will be higher at extracellular as compared to intracellular. Negative charge must be higher in case of intracellular as compared to extracellular. Alright, so that's the only thing. The action potential is caused by the rest of positive sodium ion into the cell and subsequently return to its cell to its resting potential. That's it. That's the idea. After that, let's discuss the line by line discussion. So a resting neuron is one that is not conducting an electrical signal. The change in the resting membrane potential is the key factor in the initiation and conduction of signal. And when a subsequent strong impulse or whatever stimulus is applied to a given point of neuron gates, like voltage gated channel in our muscle, like those gates in the membrane of its membrane, and opens, so sodium ion flood into the cell. The sodium ion are driven into the cell by attraction to the negative ion on the inner side of the membrane, as well as the relative higher concentration of sodium ion outside the cell. So this is very important line. You should know that the sodium ion are driven into the cell by attraction to the negative ion inside the membrane, and as well as the relative concentration of sodium ion outside the cell is quite low, so they want to come into the intracellular portion. The large sodium ion first neutralize negative ion on interior of the membrane, inside the membrane, and then goes to become positive charge momentarily. As a result, membrane potential in localized in this region from 70 millivolt internally at initial state, then the resting potential to about minus 30 millivolt in the short time, like momentarily. The sodium gate then close voltage gated like channel, which is responsible for cardiac muscle contraction. Like that, the sodium gate then close, and cell membrane quickly returns to its resting potential. This potential, this change in the potential from minus 70 millivolt to minus 30 millivolt and back to minus 70 millivolt is known as action potential. After resting potential, the sodium gets open, and then the cell membrane quickly returns to its normal potential. So that is the point. Inside the cell is minus 70 millivolt, and after positive ion come into the intracellular level, the potential difference inside is almost minus 30 millivolt. So it is considered to be resting state. After that, quickly it again open certain channel. Let's say after that, opening of the certain channel leads to again the sodium gets go the sodium gets after closing the sodium gets particularly, but other channel is open, then it usually the cell membrane quickly return to its normal resting potential. And the change in potential minus 30 millivolt to minus 30 millivolt back to minus 70 millivolt is known as action potential. Again, I think I have just mixed up some quite a bit, but you should know that after, if that's right here, bullet point, you should know that if intracellular level is negative value in high amount, it is considered as normal state. But after resting potential, which means negative value becomes low. as compared to positive intracellularly, then it is resting phase. After that, regaining again, regaining again to intracellularly as positive value more, positive value more is considered as action potential. So action potential condition is when the negative charge again has more value intracellularly as compared to extracellularly. So in those conditions we are going to say it is action potential, and if the resting potential is to be considered, then in resting potential there will be much lesser negative value intracellularly, just for momentary period of time, and after that the action potential lasts for few milliseconds, and then it is electrical signal that propagate down the axon, typically at the speed of about 15 meters per second to the next neuron or to the muscle. That is